Hi everyone, welcome to our August machine learning workshop today. Um, Thank you everyone for joining. It looks like we have a really diverse crowd. I'm sure we will have more people joining. Before we begin, just so everyone knows, so we've muted all attendees and video will be off, but please feel free to use the chat room. We will be answering questions there and feel free to interact that way. We will be monitoring it for any questions and we will ask those along the way. All right, so before we begin, for anyone who is new to Our Ladies, Our Ladies is a worldwide organization that promotes gender diversity in the R community. So we do that via meetups, mentorship, all sorts of special events. We used to have happy hours, code and coffees, but not currently, but we hope to resume those in the near future. And we do all of our events in a friendly and safe environment. So we want to be welcoming of everybody. If you haven't done so yet, please make sure to read our code of conduct, which you can access at this link over here. All right, before we begin with the meetup today, I want to do a few announcements. So we would like to, sorry, I have a lot of things popping up here. So first of all, I would like to thank our sponsors, Our Ladies Global, for allowing us to use their Zoom account and hosting this meetup therefore, and our consortium who has sponsored us for several years at this point. I would also like to tell everyone about our mentor speaker directory. So if you haven't checked it out yet, please have a look. So we have a list of we have a link where you can access people who are willing to act as mentors or as speakers, and we are focused primarily on the gender diverse population in the art community, but feel free to check it out and sign up if you are interested in being either a mentor, a speaker, or a mentee. Also, we want to let everyone know that we have a YouTube channel. So this meetup will be recorded and we will upload this meetup to the YouTube channel immediately afterwards, so within a couple of days. We also, if you don't know, we have a Slack space, so feel free to sign up and join. We have a really lively community there, and if you have any questions about R or any interesting or exciting news you want to share with the community, please feel free to join us on Slack. We have a host of upcoming events after this one. So we have in September, we will be hosting a meetup, a workshop on decision trees and random forests. In October, we will be hosting a meetup that will introduce you to Flex Dashboard and debugging with shiny objects. And in November, we will help you create your first R package in 1 hour. So stay tuned for those. We will post all of the meetup details on meetup.com on our Our Ladies Philly account. I wanted to let everyone know that there was also, I just recently found out about a book club that is likely to start soon. So this is going to be focused on the book Statistical Rethinking. They haven't figured out a schedule yet, but they are first waiting to see how many people are interested. And so go check out the GitHub account and sign up and let Faith know that you are interested in participating in this if you are. It should be really fun. One more announcement here. Wanted to let everyone know there is a community for underrepresented minority users of R, and this is called MIR community. You can find them on Twitter. You can go check them out. They do really good work. And if you want to sign up to their community, there's also a registration form. We will make these slides available as well. Before we go into the workshop, just a quick mention here. As we've done in the past, there is a Google document where we will collect all the questions and answers. So if you want to just ask a question to Jackie, just go to the Google Doc and write your question in there. And Jackie will stop at several times during the workshop and we will ask her the questions from the Google Doc and we will record the answers in there as well. But in case you, and we will post this link multiple times in the chat, but in case you miss it, it's okay. You can ask questions in the chat and we will monitor the chat room as well. All right. And then finally, just reiterating, we are recording this meetup, but please don't hesitate to live tweet about it, make some noise in the Twitter world so that we can get the word out there about the good work that we're doing here. All right, I think that's it with the announcements. So introducing Jackie, who will be our speaker today. Jackie is Principal Data Scientist at the Childhood Cancer Data Lab, which is an initiative of Alex's Lemonade Stand Foundation. She works to make large volumes of cancer research data accessible by storing them in formats that are easily mineable, widely available, and broadly reusable. And she trains researchers on how to use these formats to accelerate cures. So she does really good work there. In her past academic life, Jackie has worked on cross-platform normalization of gene expression data, on unsupervised transfer learning for rare diseases, and on developing novel frameworks for analyzing high-throughput molecular data from multiple tissues, clinical manifestations, and drug trials. So without further ado, Jackie, I'll pass the share screen over to you. Welcome.
Thank you very much for having me. I'm excited to be here and cover some machine learning topics and concepts. And thanks everyone for coming out. I am gonna start sharing my screen here, and hopefully you can see my slides. Okay, wonderful. Yeah, so I want to start out by talking a little bit about kind of the goals for this evening. So what we want folks to come away with, which is essentially an understanding of why it's useful to study transcriptomic data from tumors, why we might want to do that. Some ways we can use unsupervised machine learning to reach particular analysis goals that we may have. We hope that you're going to get an intuition for some of the challenges that we face when kind of trying to identify groups of samples or tumors, with the idea being when we want to do that, often we want to know kind of if there are similar kind of biological processes happening at the time of collection. Hopefully also getting kind of a feel for, or start to get a feel for, different dimensionality reduction approaches that we can use for visualization, and also kind of an idea of what cases we might want to use kind of more domain-specific approaches to learn low dimensional representations of our data. So what I'll say is, in general, this is kind of meant to be a crash course or kind of jumping off point, that we're going to go really pretty light on the details, kind of underlying methodologies, particularly for more complex methodologies. But along the way, what I try to do is link you to the material this is adapted from, which is generally training material from the work that we do at the Childhood Cancer Data Lab, or kind of tutorials or other references, so you can kind of continue your learning journey, if you will. Yeah, so we'll go through. We'll be a little light on kind of some of the details. We'll link out where possible. And what some of the assumptions with this material is that folks will have some familiarity with R, RStudio, R Markdown, the tidyverse, things like navigating directory structures with file path, and then some familiarity with the concept of clustering or finding essentially cohesive groups of samples. The other thing is, we're gonna try, this is my first time in such a setting using RStudio Cloud. So hopefully that will go well. If you would like to follow along with the notebook, you'll need to create a free account, and basically we'll have a link at the end of the slides that will allow you to create a project and have the notebooks that we'll use. The other thing I want to do is acknowledge the CCDL team. So I mentioned that these materials are generally adapted from some of the work that we do at the CCDL. So the science team in particular, Josh, Candace, and Shantae have had a big hand in putting parts of this together that I'm gonna show you today. So although I'm gonna be the one presenting, you know, these folks are behind the scenes as well. So it's important to acknowledge them. And the other thing I want to highlight is part of what we do at the CCDL is creating products for researchers with different expertise. So we love to hear from folks about their experience with data and collaboration. So Deepa is our user experience designer. She would love to talk to you about your experiences. You can follow this link. I think the PDF of the slides has been shared in the chat, it looks like, or you can also reach out to her on Twitter. So we appreciate any time that you might be willing to spend with Deepa so we can understand your kind of processes and experiences a little better. So before we jump into the notebook, I wanted to provide a little bit of background. So machine learning, kind of a really basic one-sentence introduction for what it is essentially having a computer program learn to perform a task, for example, predicting an outcome, rather than kind of explicitly programming things like rules to accomplish that task. And kind of at the most basic level, there are two kind of classes of machine learning algorithms. We're given a data set, we can ask what patterns exist in this data. So if we have two kind of dimensions or features that we're measuring, and each of these is a sample, we can do things like ask if these samples form clusters. And in this case, asking a question like what patterns exist in these data, that is an unsupervised machine learning problem. On the other hand, we can also say, given I have some labels, so I know that there are two groups of samples in my data, what fits this pattern? So we'll do something like train a classifier to basically draw some decision boundary between the two classes and predict class labels, for example. And this is supervised machine learning. So what I'll focus on today is unsupervised learning. So we're gonna cover some of these questions. So like which samples are most similar to one another in my data, or observations, if you will. How many groups of samples exist in my data, which is, we'll show you one way to look at that, but that is, as it turns out, a very hard problem. And also questions like what patterns of gene expression exist in my data, how do the genes vary together? So before we get too much further, we're going to use gene expression data or transcriptomic data. I'm going to probably somewhat incorrectly kind of switch between the two. But what is that? Because, you know, not everyone might necessarily have the background. We're going to have a little bit of a digression that involves cupcakes. Yeah, I don't know who first introduced me to this analogy, but it really resonated with me, and I hope you enjoy it too. But the central dogma of biology goes like this. You have your DNA, which gives rise to RNA, which gives rise to proteins, and then the collection of protein products together, along with some other things, kind of come together to form cells and tissues. So I like this for a baked good based kind of analogy. Is the DNA is kind of like a cookbook. So it's a set of recipes that could possibly be made. The RNA is a single recipe itself. So this is what's going to be made. The protein's the product. So going from your cupcake recipe to your cupcake. And then everything together, what the recipes can give rise to, those products together form things like cells and tissues. So you can also think of this kind of as like a bakery. So what we're actually going to look at is if we assume every time we make a copy of a recipe, we're going to get a product. We can kind of measure the collection of recipes at the time when we collect a sample. It's a little more complicated like that than that, but for the most part, this is how we kind of think about this. So when we're measuring what we call genome-wide, so you can think of it as cookbook-wide, gene expression data, it's kind of like taking a peek at all the baked goods being made in the bakery at the time the recipes were run. So that's what we have there. If there are more kind of copies of this recipe or recipes that kind of participate in the same process, we infer, hey, this process would be active, and it's kind of giving us a hint into what's happening at the time of sampling. So what's the data actually look like? It's a little less exciting than cupcakes, I would argue, but here we are. So how this is typically stored is that we have genes and we have samples. And in humans, we're going to be looking at human data today. And so that will usually be tens of thousands of features. So what a row will be in this case, there are features. It's a measure of the expression of the RNA, the recipe, for all the samples in our study. And a column is going to be the collection of expression values for a given sample or observation. So kind of taking all of the collection of recipes together, looking at their levels, we can kind of have an idea of what's going into that sample, although it's not perfect. But that's the general idea. So when we're using this data, often in like a tumor setting, what we're hoping to do is kind of get an understanding of the biology happening at the time of sampling and kind of group samples together. So why might we care about sample-sample relationships? So I am kind of pointing out an early paper kind of in the gene expression field looking at molecular subtypes of human breast cancer. And these have been defined. This has actually gone on to the point where this is it is included in kind of a diagnostic test. And often if we want to find samples with different underlying processes and kind of make a statement about them or kind of take them for further study, we also want to know often kind of how stable those groupings are going forward. So that is often, when we're looking at things like tumors, a question of interest that we have. We also kind of often want to use dimensionality reduction to explore the structure in our data. So I mentioned we're going to be working with human samples, which is tens of thousands of genes, which can be really hard to visualize. So often we want to kind of collapse things into lower dimensionality, using some techniques you may be familiar with, like PCA, and some newer techniques like roticity, all of which we'll kind of play around with a little bit today, and kind of see if we can see structure that corresponds to, ideally, biology. Some cases, not so much. Sometimes it's some kind of technical effect that we're not particularly interested in measuring, but can kind of arise nonetheless. So those are kind of the first questions. What samples are most similar to get to another? We can do that through clustering. We can do different things to kind of test the stability of groups and see how many, what's the optimal number of groups. Turns out that's kind of hard. And we can also ask things like what patterns of gene expression exist in my data and how the genes vary together. And often, for me as an analyst, this is what I'm kind of interested in. And the reason we can go from our gene by sample matrix to something that is lower dimensional, where our patterns or latent variables are often biologically meaningful, or we're hopeful that they will indicate something, for instance, like a cell type, which tends to be kind of like a linear signal a lot of the time. We can do this because individual genes kind of are coordinated in their expression. So kind of genes, these individual features are not working in isolation. They work together as part of kind of molecular processes or pathways, which are kind of put together as gene sets. So often, my goal as an analyst is to go from this to kind of a lower dimensional space where hopefully the measurements of this kind of combination of genes is kind of may tell me something like the presence or absence of a cell type, which is often kind of a question I'm after, because a tumor or kind of any kind of what we call a bulk sample will be usually a collection of all different kinds of cell types in some cases. So it can help us kind of figure out what's going on at the time of biopsy. So at this point, we're ready to kind of move over to RStudio Cloud. I'm gonna stop sharing and we'll get.
Question, Jackie. So the space, we were able to connect to it, but it's empty. Is that the plan?
Yes, that's a-okay.
Great.
So I'll start sharing my other screen in a moment here. All right. So if I go and log into my other account, so you'll see what I see, or we'll see the same thing. So I'm going to say join space. So this should be what this looks like kind of for you as a participant. Welcome. If we go in projects, no project, that's okay. I think that's what Carla was alluding to. So what we can do, we're able to say new project. I'll take a moment here. Okay, so you should see kind of your files pane in RStudio Cloud be populated with these two notebooks, and this also should have all the dependencies, fingers crossed, and all of the data files that we need. So I'm gonna go ahead and X this out here. I'll just call this workshop. One thing I did want to note is that I'm gonna get rid of this workspace as soon as we are basically as we wrap the workshop up, but you will have access to these materials on GitHub. They're the same as we have here. And the other thing is we also provide a Docker image if that is of use to you, that has the dependencies needed for the workshop to make that perhaps a little bit easier. But yeah, so hopefully this is now, we're taking a look at the same thing. One kind of Zoom note, which folks may have already picked up on, but if you intend to kind of follow along, you'll probably want to exit from full screen right now. often the default for Zoom is if I am sharing my screen, it takes up all of your screen. And if you'd like to follow along, I'd kind of recommend stopping that and kind of putting these next to one another. But yeah, so we're ready to kind of get into it. And we'll start running things. One thing I'll note, if we were in person, there would be kind of more live typing interspersed here. Given the format, we're not gonna do that. So, you know, if you are maybe a step behind, you will not fall as far behind. All of the code is entered in here for you. Yeah, so we'll get into it. So we're going to use RNA-seq data. That's kind of the newest technology, or one of the newer technologies for assaying the gene expression data that we kind of use the cupcake analogy for. This is from a project called the Open Pediatric Brain Tumor Project. We're not going to create a heat map. That's not true. But OpenPDA is a collaborative project. It's organized by folks at the CCDL and also D3B, which is the Center for Data-Driven Discovery in Biomedicine at CHOP. You can read more about the project here. So this may be of interest to you, but this is on GitHub. You can access data and also kind of participate in this open analysis of these data and manuscript writing on GitHub. So I encourage you, if that sounds like it's of interest to you, to check it out. What we've done, we have the publicly available data. I've done some transformation and kind of filtering ahead of time in preparation for the workshop. And we have a setup folder here. This is also, again, available on GitHub that you could kind of see what was done and a little bit more information about that. In this particular workshop, we're going to be using medulloblastoma samples. This is the most cancerous brain tumor in children. And it's worth noting there are 4 kind of subtypes that have been identified, which we'll come back to in a moment. If we're interested in doing things like identifying groups or clusters in expression data, we're also often want to kind of validate those in some way. One question of interest is how many groups are there, or kind of checking for agreement within clusters. So in this particular example, we're going to take this data from the OpenPDA project, demonstrate a technique for kind of cluster validation, and also look at how these clusters overlap with sample labels, because there's more than one way to validate a cluster. I'm only going to show you kind of 2, but we link out to kind of a course that will go into more depth. So we're ready to start kind of running these chunks, which we can do by hitting this play button. And that will load in our libraries for us.
Jackie, let's just pause for a second. I think some people are struggling to connect to the workspace. So
Gotcha.
Let's give people a few minutes to work out the details.
Yeah, I think the key is that you have to be in the correct space. So follow the link to that space, 82434, and then create the new project. If you create a new project in your space, it will be empty. And it will look like a new RStudio session. And then if you look at the files section, you should have those new files.
create a new project here. I was in the workspace. I went to projects and then
Yep, so this looks like a new session, and then you have the files over there on the right, the two markdown files.
Okay, I think some more people are starting to get it.
All right. If people are following along and have access to the yes and no buttons here, mark yourself with a yes and no if you got it to work. And we will try and troubleshoot. I think we can proceed otherwise.
okay.
Yeah, let's proceed and we will troubleshoot on the side.
Okay. All right, so I'm in a new session. So I'm gonna run this again, load in these libraries. Set up my file path. So I have, like I said, prepared data ahead of time. That is in this data folder here. So I'm gonna go ahead and run that. And then we're ready to read that in. So these have already been transformed using a transformation called variance stabilizing transformation. There are kind of certain properties of this kind of data. What we've done is made the variance less dependent on the mean values. The values in this case are kind of influenced by factors like the length or size of a gene. But following this transformation, the values should be on a more similar log to like scale. This is a kind of transformation that's recommended when we're going to be doing the kinds of things that we are doing today, like clustering or any kind of working with distances. So I just wanted to kind of note some things about the way these data are stored. So gene expression data are typically stored where rows are genes or features and columns are in samples. That's kind of not, I would say, using a lot of other applications where rows are going to be your samples and or observations, and your columns are the features being measured. So I'm going to use an example from the Palmer Penguins package. If we take a look at the head of this, we can see each of these rows is an individual penguin, and then we have features like the bill length and depth, body mass, etc., are stored in our columns. If we take a look at our RNA-seq data frame, we can see here are our genes. So this is generally the features that we're interested in, and then each of these columns is going to be a sample. Sometimes we're going to want to transpose our RNA-seq data such that the features are columns and rows are samples when we're working with kind of generic packages that would take any kind of numeric matrix. We're using a package called Consensus Cluster Plus in this particular notebook. That's kind of explicitly designed to work with transcriptomic data or gene expression data. And this is the orientation that it expects. So quoting from the package documentation here, a data matrix, it expects the data to have the columns as items or samples, and the rows are features. So for example, a gene expression matrix of genes in rows and microarray in columns. So that is what we have in this particular case. The other thing you may have noticed about the penguins data, if we take a look at the head again, is that what I would call kind of categorical variables, or kind of, I think of them sometimes as labels, such as the species and island and such. This is kind of stored alongside the numerical data. But when we're going to store our gene expression data in a tabular format, usually the sample attributes, things like disease type or any kind of sample label, is stored separately. So we read that in something called clinical DF. And then we can see we have our sample identifiers here in this column. And then we have things like our labels of interest, things like descriptor of the tumor, molecular subtype, and such. So our columns, again, in our RNA-seq data frame are samples. Our first column is going to be our gene identifiers. So we can check if our samples are in the same order in both of these by running this step. And we can see that they identical returns true, so they're in the same order. In this particular case, we're going to use a matrix for our next steps. So we'll convert our gene symbol or gene identifiers to our row names and then make a matrix. Okay, so we are going to attempt to identify groups of samples in our data. And the idea is by comparing, if they have kind of similar expression profiles, they may have similar underlying kind of biological processes at play, if you will. The particular method we're going to use is something called consensus clustering. And the intuition here is that consensus clustering aims to find the consensus across multiple runs of an algorithm using a resampling procedure. We're going to use a particular package called Consensus Cluster Plus. This was first introduced in this paper. We're going to take a brief look at some simulated data and results from this publication so we can look at kind of an ideal case, and then I'll show you the not ideal case, which is the real data that we're working with. So what consensus clustering outputs is going to be something called a consensus index. And that tells us essentially how often two samples are clustered together over multiple runs of the algorithm. If it's 0, two samples are never together. If it's 1, samples are always together. And these values can be visualized in something called a consensus matrix. So what we're showing here is a consensus matrix from a simulated data that contains, we know that it contains 3 groups. Because this is showing us the relationship between samples, that's what the consensus index tells us. Both the rows and the columns are corresponding to samples, and then essentially a cell is going to be white if the consensus index equals 0, and red if the consensus index equals 1. So what this is telling us here, these values are all 1 along the diagonal here. Samples in this group always cluster together, and they never cluster with these samples. Samples in this group always cluster together. They never kind of cluster outside of that group, and so forth. And there are kind of other ways to look at the consensus index values that we get back. So we can look at the density. And kind of ideally, you want a really clean consensus matrix, right? If we want to say these are kind of really solid groups of samples, we'd want this to look clean like it does with this simulated data, because it tells us that between runs, these kind of never venture out of their cluster. So we expect these to be pretty stable. So we can look at this using this density plot. We can also kind of look at the CDF. And one way to kind of interpret how many clusters you may have is to do things like look at the relative change between kind of different K, because we're going to specify multiple K often with consensus clustering, kind of force there to be that many groups, and then calculate consensus index values. So often in this case, when we go from 3 to 4, we no longer see an increase in the CDF. And then in this case, usually you can make, if it looks this clean, we would say that there are 3 clusters. Of course, this is simulated data, so we know this to be true. We can also look at a simulated example where there are 5 groups, and we're looking at kind of multiple runs going from 2 to looks like 9 K, setting K to those values. And we can see, if we look at the consensus matrix for K equals 4, and we know there's 5 groups, and then we also look at that at K equals 5, we can see that this looks a little bit cleaner, right? So there are kind of, when we lump kind of all of these together, this looks kind of less clean than this. So basically what we're after intuitively is to kind of look for a clean consensus matrix. And then things like the CDF also tell us a little more info. We can see there's a difference between 4 clusters here and 5 clusters. Okay, so this is one way to determine the number of clusters in your data. It was kind of, this package is specifically designed to look at that in data that we have here. I'm linking here to a cluster validation essentials course, which kind of is more, has more methods than we'll cover here. But what we can go ahead and do is run this, and this will spit out a bunch of plots at us. So we'll have from 2 to 15, because we're setting max K here, we're going to have a consensus matrix for all of these K values. So I can go ahead and hopefully go through. We probably have one sample that's kind of an outlier here, if you will. And we can kind of just step through and take a look. Okay, so let's say the difference between 5 and 6, this looks quite a bit cleaner. If we go to 7, things kind of look less clean again. 8, 9 kind of starts to look clean as well. And so on and so forth. Things starting to break apart again.
Jackie, maybe we can take a quick question break.
Sure.
I've noticed there were a lot of questions that people were asking. So one of the questions, are each of the samples a different patient, or are they samples from the same patient in your data set?
Yeah, so the majority of these are going to be samples from individual patients. There may be some cases where they are not kind of independent specimens. They'll be from the same patient, but that is not super frequent, and I don't know about this kind of disease type specifically in this data.
Uh-huh. Okay. How is the reduction of dimensionality through machine learning different from K clustering?
So often when we're doing, so there are kind of different ways to do dimensionality reduction. So just saying dimension reduction is, we're just going from fewer dimensions. So there are ways to do that where you're doing something like feature elimination, because, so we'll see in the next notebook, one way to do that is basically seeing what features have high variance and kind of removing things that don't have high variance because they're unlikely to tell us about the structure of our data. That is a form of, that is reducing dimensions, but there's also ways to kind of do feature extraction where you get kind of a new set, from your input data, a new set of variables. So something like PCA, right, you're transforming your data to have kind of new variables that you're then visualizing. K, clustering, often I think how I would use that would be on the original features themselves. But I might do a step where I do some kind of feature elimination or feature selection.
Okay. Let's see what else. Alice, want to jump in and help me out? I'm trying to sort through all the questions here. And
I've been watching them. There's actually a question more on like your workflow at CCDL. There's a question on, you're part of a 5-person team. And so your R development workflow, do you all push code to a Git repo, or do you use RStudio Cloud? How do you coordinate when you're collaborating on a project in a team?
That is a great question. They have all been great, for sure. Let me just say that. But so that is, depending on the project a little bit, it'll vary, because I don't think any strategy is one size fit all. But what I will say is kind of each project that we work with, we're trying to move to a strategy where each of them have contributing guidelines. So we do use GitHub extensively. You can find open PBTA on GitHub, but that should lead you to the Alex's Lemonade organization where you'll see like our training repository. But we branch, we use and work on our own branches. We have a pull request model for everything that we do. And we often will use Docker as a big part of our workflow for certainly a project we're working on now. But you know, depending on what's going on there, sometimes pulling from S3 will be involved. But yeah, use GitHub. We use a branching kind of model of development. Everybody's code gets reviewed, and we often use Docker. It does a lot of things we work on. We use Docker images from the Rocker project, so you will essentially have our studio server running in a container. Yeah, we do our training workshops on an RStudio server Pro now that we host on AWS. I hope that answered that question. That's kind of the general idea. If people have specific questions about how to organize a team of people to do analyses, I have certainly gotten more experience with that kind of lately with OpenPBTA, and I'm happy to chat more about that.
Okay, maybe one more question before you get back in, because it's, I think, related to what you're showing now, which is, are there metrics for how, quote, clean these clusters are? Like you said, when you went from 4 to 5, it looked cleaner, but how could we make a metric or something on that?
So that's kind of what this is doing for us here. Looking at the density, and then kind of looking at once we go from 3 to 4, the area kind of doesn't increase more. So we see this kind of drop. I think realistically, when we're working with data that's not simulated, that is not so clear. So we'll look at that in a second. So we can actually just kind of jump into the delta area plot that we got for this. We can see kind of like, maybe you could argue for 6 because there's not a big increase between 5 and 6. But then we go back kind of up at 7 and then kind of level out at 9. And that is kind of a way using that CDF, using the delta plot can help us quantify the cleanness. But there is like I don't want to gloss over the fact that there is a fair bit of just kind of looking at this by eye. And if I were to do this where I really wanted to be sure about my stability of my groups, I would probably use multiple methods. One other thing I just want to point out is this produces something called a tracking plot, where it allows you to look at how kind of samples cluster together over multiple K values. And then we can see once we get to 9, we kind of have 3 main groups that emerge until we get to 12. And then we can see that by eye too. So before 9, if we look at K equals 6, 7, 8, 9, we see that there are kind of like 3 groups, and then eventually it'll start to break apart and look more like 4, which we can kind of see that here as well. So we can move forward with 3 main clusters, but I do want to emphasize you could probably make an argument for the 2 groups that show up at K5 through 8. If we look at this tracking plot, because, you know, ideally we'd see something like we see up here where it's really clear that there are some number of clusters. But again, if I were to do this and it was really important that I was sure about the number of clusters, I would use multiple kind of different methods. So we're going to move forward with these K=9 labels, because to me, right, we don't see kind of a spike after the fact here. And we can take a look at kind of the table of those results. And what happens in a lot of these cases is that we're kind of getting groups of 1 or 2 samples, which is not necessarily that helpful for reaching our analysis goals, where we're saying here are groups of samples that have kind of maybe similar processes occurring. If there's 2 samples kind of off, that might not be that helpful for our analysis goals. So that's another thing to keep in mind. It's one thing to kind of try and pick the stability of our groups and say how many there are, but it also is always going to depend on kind of the question that we have, and having a cluster of 1 in a lot of cases is not going to help us reach that analysis goal. So what we can do is for K=9, extract those labels. I'll pull this over here. And then one thing that we can do is check using kind of external labels. So I mentioned at the top of the notebook that there are kind of multiple molecular subtypes in this cancer type. The subtype classification, what I didn't tell you at the beginning, it largely is going to rely on the gene expression data themselves. So this is the code for the classifier. So the classifier is trained to predict the subtype labels. This is an example of supervised machine learning. And we have this in our clinical data frame here. That's what's in this molecular subtype column. So we can ask how the subtype labels from the classifier stack up to the clusters we identified using this completely unsupervised method, right? We simply asked what groups are there and how stable are they? How many might there be? Again, there are lots of different ways to do this. So first thing we're going to do, we'll, from the clinical DF, pick the identifier column and the molecular subtype column. And then we'll join it with our labels from consensus clustering for k equals 9. And then what we can do is make a table for that. So one thing I want to note, we have two groups, two subtypes that look like they tend to group together. That is consistent with what we expect. These two subtypes tend to be more similar and kind of more difficult to distinguish from one another. So this big group, this big kind of cluster 1 in this case, tends to have samples from group 3 and 4. Our SHH samples are in cluster 3, and then cluster 4 looks like it might be our Wnt samples here. So the subtype labels, again, are derived from the data that we've clustered here. They largely agree with our unsupervised results, but it's not perfect by any means. In group 1, sometimes we'll see Wnt samples, for example. It is worth noting, though, what the classifier actually uses are different features that are derived from these data. So it's a little bit different from what we've covered here, but we might expect them to agree. And for the most part, they do. So in the next notebook, what we're going to do is things like UMAP and kind of color points by our subtype labels and take a look. I'm going to hit session info. Might be a good time for questions. While I save this.
Yeah, we have a couple of questions. So one about this actually. What do the counts correspond to in the contingency table? So what is 76 in k equals 1 and 1 in k equals 9?
Gotcha. Great question. So that, yeah, skimmed over that there. This is the number of samples that are in that cluster. So there's 25 samples in cluster 3. There are 11 in cluster 4, and then we have a bunch of that are 1 or 2 samples. The big one is cluster 1 here. And then when we take a look at the table here, we can see how those, it was 76, I believe, samples that are in cluster 1 are spread out across our subtypes, our external labels in this case.
Gotcha. Thank you. Let's see.
I'm going to go ahead. Sorry.
Oh, Alice, any unanswered questions in the Google Doc?
So the one that might be hard to answer because it was a continuation of the question that you answered in the last break we had, which was around, different methods of dimensionality reduction. Mm-hmm. And I think you had mentioned something about dimensionality reduction based on variance.
Yes.
Can you just repeat that? We didn't catch it.
Why don't we hold that to the next?
Perfect. Okay, stay tuned. It's a teaser.
Yeah, stay tuned.
Okay, I think that's it. The other ones were answered.
Great. So what I'm going to do, I'm going to just kind of restart. And then we can go ahead and open 02 here, which we're going to do a little bit of dimension reduction and representation learning. A very brief tour, but there are some resources that I like a lot, linked at the very end of this notebook for you to kind of continue playing around with this kind of thing. What we're aiming for this particular notebook is starting to understand how to build intuition around some of these methods. So briefly, cover some concepts. Dimension reduction, sometimes called dimensionality reduction. I will probably say it both ways during this workshop. I think I already have. Is going to be any kind of technique that reduces the number of features or dimensions in a data set. In our case, a dimension in our high dimensional space is the measurement of an individual gene. For our RNA-seq data, dimension reduction can come about through feature elimination or feature selection. So kind of what we can do is remove features or variables if they're unlikely to give us meaningful information. So this is going to be dependent on our question, as we can imagine, but some cases we'll want to remove them if they are redundant with other variables. Or we can use something called feature extraction, where we're going to form kind of new features from the input data. And that's mostly what we'll cover here. And kind of the idea or intuition behind it for this particular case, as we'll cover it in this notebook, is we want to transform our input data, which is very high dimensional. We have tens of thousands of features, into a low dimensional representation that ideally is going to retain some meaningful structure. So this is kind of on the feature extraction end of things. So often if we have a question like, what's the overall structure in our data? And does it, for example, correspond to our molecular subtypes? We'll want to go from this high dimensional data, which is really hard to visualize all of these dimensions, to something that's fewer dimension. Often we want to make a 2D scatter plot of the first 2 dimensions, where each of these points is now one of our samples. The idea behind something representation learning, which is also sometimes called feature engineering or feature learning, it's basically any technique that's going to automatically detect representations from our input data. So for example, an unsupervised representation learning method would be something like matrix factorization. We'll show kind of a specific matrix factorization method here. Something certainly like an autoencoder is representation learning. But it is worth noting that supervised methods can be considered representation learning too. For example, things like supervised neural networks. And to kind of tie this back to our slides at the beginning, often what I want to know as an analyst is if I can find a low dimensional representation where the latent variables that I've essentially extracted by doing some kind of dimensionality reduction can tell me about things like cell type or a particular biological process that might change in response to treatment, for example, or be different between subtypes. You can also use representations in kind of a semi-supervised manner, where you're going to learn the representations from the collection of data without any labels, encode a low dimensional representation of data with labels that you can then use for prediction. So there are, representation learning kind of comes into play in lots of different arenas. So we can get started. We're gonna set a seed here for reproducibility. We're gonna load, whoops. We're gonna be patient and wait for that chunk to run. Hopefully, so we've loaded in our libraries here. We're gonna look at UMAP artisany. We're gonna read in essentially the same files we had earlier, but there's a model called, from a method called plier, which we'll talk a little bit more in detail. That takes a little bit of time to run. So for the purpose of this workshop, I have done that ahead of time. And that you can see in setup, and again, that's available on GitHub. So let's go ahead and set up our file paths here and start reading in our data.
While this is running, someone had a question why you took out categorical variables instead of having dummy variables.
Yeah, so I would say part of it is generally this is how that data is analyzed, which is not a satisfying answer. But usually we kind of keep our sample labels or kind of categorical labels separate. Kind of look at what we're most interested in measuring at first. And when I say we, I kind of, my personal worldview to a certain extent, kind of look at the extract patterns from the transcriptomic data or gene expression data, and then we'll do things like check for agreement with those sample labels. I would say that is a somewhat standard path, which is maybe not a satisfying answer, but it is a truthful one.
And one more, if you can talk a little bit more about what a latent factor is before we jump into this.
So a latent variable, we're calling it latent because we've learned it from the data. It's not kind of our input feature that we've observed. I will touch on that a little bit when we get to Plier. I will touch on that in a little more detail. But the idea is we haven't observed it. We're learning it.
Sounds good. All right, that's all the questions for now.
Okay, so great. In this particular case, we do want to transpose our matrix. And now we are going to do some feature elimination or feature selection, arguably. We're interested, what I'm going to talk about when I'm doing something like an exploratory data analysis, I'm often interested in the overall structure of the data. So how samples are kind of grouping together. Is that something that I would expect based on biology? So often I have external labels. In this case, these are subtype labels, but often maybe I'm looking at an experiment where I have multiple tissues. And those tissues kind of have very different kind of protein products that they need to make a kidney and a blood. And we would expect them to kind of separate in low dimensional space, for example. Or is it something undesirable, like some kind of technical effect that is kind of showing up as the overall structure? So what we'll do is kind of eliminate features that don't vary within themselves, because we think, they're less likely to contribute to the overall structure in our data that we're most interested in. Also, given kind of time, resources, things like that, if we're just trying to get an idea of our structure, we can do things like take the top 10% of features, which are now columns in this case, and then kind of use that as input to our dimension reduction or dimensionality reduction techniques. So that's what we're doing here. We're gonna stick with the top 10%. I'll just say, often when people are using packages that are explicitly designed to do something like PCA or UMAP or TSNE on transcriptomics data, or kind of a version of transcriptomic data where instead of samples you have individual cells, what this will do this under the hood, where it's looking at kind of high variance features only. So this is kind of a common way to approach this problem. So we can read in our clinical data. And again, for the most part, we're only going to use our molecular subtype information. Oops. So now we have our subtype data frame. And now we're going to just start making some plots, which is my favorite part, one of my favorite parts of an analysis. And here we're like definitely not going to get into the details. We are trying to build an intuition by kind of making visualizations. So the first thing we're going to do is principal components analysis. it's a dimensionality reduction techniques that's going to capture the main sources of variation in our data, kind of our maximum variance in the first two principal components. PCA essentially applies linear algebra to find the axis of greatest variation in the matrix, then the axis perpendicular to that with the most remaining variation, and so on and so forth. So what you end up with are these kind of new variables that are linearly uncorrelated. So we're gonna go ahead and run PCA. We're gonna add our subtype information to our first two PCs, and then we're gonna make a scatter plot. And we're gonna color those points by our subtypes. Okay. So we have kind of our first principal component, which captures most of the most variance. Principal component 2 will be, again, the axis that is has perpendicular to the first one that captures the maximum variance, and so on and so forth. Some things I notice about this is that these are kind of points kind of fill all of the space, but these are not necessarily, the results that we see here are not necessarily kind of different than I would expect based on the consensus clustering results. So we see group 3 and group 4 kind of close together here. Kind of that principal component 1 kind of separating that from the kind of bulk of the samples in the other two molecular subtypes. And that, to me, the fact that we have kind of these SHH samples kind of grouping in, or they're close to group 3 and 4, that kind of has alarm bells kind of going off for me a little bit, because I would expect, and partially like I'm quote unquote cheating, where I have like some basic understanding of the literature for these subtypes, but I would expect kind of group 3 and group 4 to be kind of close to one another, not necessarily obviously distinguishable on a graph like this. And then our WINT and our SHH for samples to kind of be clearly their own clusters. But what can happen here is sometimes this produces plots where kind of points are across the whole plotting space. That gets more pronounced as you have lots of samples often. And now there are newer methods, particularly if we have like thousands of samples, or looking at where using single cell transcriptomics, where again, we don't have kind of samples, we have individual cells that we have as our observations. This can be kind of an issue to see, to recover kind of well-separated clusters. So that is where newer methods like uniform manifold approximation and projection, or UMAP. So what this is a machine learning technique designed to provide more detail in highly dimensional data than typical principal components analysis. PCA assumes that we care about the variation has like a particular distribution. UMAP allows us to kind of learn more complicated distributions. And we're not going to go into the underlying mathematics. That is not my wheelhouse, I'll say. But here is the paper that initially described UMAPs. One of the advantages, or the main advantage of the change in underlying assumptions, is that UMAP can do kind of a better job of separating clusters, especially when those clusters are kind of more similar to each other than other clusters. You may have also heard of t-SNE, which stands for T-distributed stochastic neighbor embedding. It has similar properties to UMAP, and often in my experience, we get similar results. I will say in specifically the application to kind of high dimensional biological data, there's ongoing debate about which of these two techniques is kind of superior, and whether that has to do with the underlying algorithm and implementation or kind of the parameter initialization defaults. You know, when they're often, to get a sense of our structure data, why that happens is not my main concern. But UMAP seems to produce kind of slightly better results, and sometimes it runs a bit faster. So what we're gonna do is run UMAP. This performs PCA first as our first step, uses the first 50 principal components, which is basically often recommended for TCNE applications. This is one of those things where it's a rule of thumb, but it's not guaranteed to kind of be the best for your application. But what we'll do, we'll run UMAP, we'll take a look at the coordinates, we'll get 2 dimensions out of this, and then we're going to color the points by subtype again. And then we can take a look. So one thing that you may notice, we get kind of more well-separated clusters. We kind of have this emphasis kind of more on local structure, I would say. But we still, this has not resolved kind of the thing that gave me pause when we looked at our PCA plot, where kind of some samples, I would expect these green samples here to be over with these SHH samples over here. We have kind of some group 4 samples over here. So part of this being kind of a project conducted in the open, I was developing some training material using these data and the sample labels, and I, was able to file an issue, and we are kind of currently taking a look at why that might be. So I would say stay tuned if you'd like. Well, hopefully next week I'll get into that a little bit more and may have an answer for you. But it is a good, perhaps, lesson in visualizing your data and seeing if things are as you expect or not. So that was using default parameters. We're gonna now do a bit of experimentation with a particular parameter called the N neighbors, which is the number of nearest neighbors. What this does is constrains the size of the local neighborhood size when learning the manifold structure. So how to think about this, lower values are going to emphasize the local structure of the data versus the global structure. I've written a wrapper function that will allow us to experiment with this. And then we can just kind of run this several times and see what happens. So if we set a different seed, this is going to be the kind of default. We've already run UMAP once using this parameter. And we can see the results are different. So that's something to be aware of. It'll be different using the same parameters across different runs. But if we're kind of just looking at the overall structure and we're not necessarily using these values, we're just using this for visualization, that is okay. We'd be a little more concerned if we were going to use these kind of downstream. One thing I will say is if you are going to kind of move forward with a particular N neighbors parameter, you might want to run it multiple times and make sure you're not getting anomalous results. And I think that's kind of a good rule of thumb. Run things a few times and make sure the seed that you landed on in the first place doesn't give you results that are wildly different or would have a very different interpretation from kind of other runs. So let's try a variety of values and just kind of see what happens. So we'll start with 3, which should emphasize local structure. And we can see things are kind of, we have smaller clusters. Or kind of, they look, I would say, tighter here. Then we can kind of go to 30, see what that looks like. And that kind of looks a little more diffuse, kind of a more emphasis. We're going back the other way, where we're kind of more interested in, we're emphasizing global structure a bit more. And then finally, what we'll do is 50. And I would say interpretation-wise, right, we're not getting values that are wildly different. You know, our plots don't look that different. We might kind of grok or kind of get that there are probably about 3 groups in here, and they largely correspond to our subtypes. And I'll say for this particular kind of histology, we kind of expect this to be really big signal in our expression data. And then the last thing we'll do is run t-SNE. It's another nonlinear dimensionality technique that can produce well-separated clusters. We're going to use perplexity, which is kind of similar to n-neighbors for UMAP. We go ahead and run that. We can see we get, I would say that that's pretty similar to this here for this particular kind of analysis task that we have. So some really brief takeaways. Newer methods that can like learn these nonlinear patterns, they can help us find well-separated clusters, which can be really handy for visualization, particularly if we have a large number of samples that we're kind of interested in working with. The results, though, ultimately depend strongly on parameters that balance local versus global structure. So partially you want to pick the parameter based on the question at hand, and just as part of like getting an intuition for parameters, it can be helpful to select multiple and kind of see how the visualization changed. And I would encourage you to do that. One thing to be aware of is that the interpretation of these methods may sometimes be counterintuitive, because the distance between clusters may or may not be necessarily super meaningful. What I will say, if I kind of skip to the end here, where we have multiple resources that are available, this particular example has a lot of great visuals that will allow you to play around with t-SNE, and we'll kind of go over some of the potential pitfalls. This is great for that as well, covering kind of the pros and cons of different dimensionality reduction techniques, more so than we'll cover here. But hopefully you can kind of get an intuition for how these might compare and an idea of how you might go about kind of playing around with these with your own work. And the last thing we'll cover is something called Plier. And we are going to have kind of a whirlwind tour, as we have been throughout the workshop, of this particular technique. So this is PLIR stands for Pathway Level Information Extractor. It is specifically designed to work with gene expression data. Everything that we've talked about so far can be used with kind of any set of data. Just because you can use it doesn't mean you should, right? It depends on your question and whether you do need to do dimensionality reduction. But this one is specifically designed to work with gene expression data and has certain constraints that make it useful for the questions that I'm often interested in, where I'm trying to learn kind of biologically meaningful patterns. So we put this in the context of kind of where we started, where we have our gene by sample matrix, and we want to kind of go and get these measurements, these latent variables that are a combination of genes. And we get latent variable expression values for each sample. We're gonna have, PLIR has certain constraints such that there's gonna be some preference for pathways. So pathways are typically sets of genes that have been often curated by domain experts that say participate in a similar process. So we can use that kind of prior information in a form of genes that we know in some context will work together to guide what latent variables we get out. So I will briefly cover why we like PLIR. We can get a low dimensional representation of our data. That's often really handy for doing things like testing for differences between groups. It can be kind of nice to have less features that we're looking at. What PLIR specifically is going to learn is correlated patterns of expression in our data, and what it gives us back is these latent variables. If you are familiar kind of working with these data, the authors say that this learns kind of an eigengene-like combination of genes expressions. It is called latent because we're not directly measuring it, but we're instead kind of inferring this from our individual gene measurements. And it's going to include penalties that some of the latent variables are going to align with gene sets we give it. So they're going to align with kind of coherent, hopefully, biological processes, so we can use those to answer kind of biological discovery questions that we may have. The other thing is that can be handy is that one thing that's of interest, particularly kind of in the cancer domain when we're looking at tumor samples, is immune infiltrate. So how many immune cells, what they are, kind of their identity or properties of those kind of cell types can inform us often about the biology largely, and what that can be really, that's an area of interest in tumor samples specifically. So if we give PLIR gene sets that correspond to particular immune cell types, we can get latent variables that kind of give us an idea of the proportion of those cell types, immune cell types in our samples. The other thing that's nice is not all of these latent variables will necessarily align, and in practice, not all of them do align with the kind of cell types that we give it or the coherent biological processes that we give it. Some of them are instead gonna capture unwanted technical variation that we're not interested in, like when the sample was run. That's often not what we're after. In experience, kind of practical experience with the method, it does this pretty well. So kind of keeping biological signal is in some latent variables, others that don't necessarily align with kind of input that we give it will maybe capture something like what we would call a batch effect. I am, in the interest of time, not going to go through this figure. I will say if you are interested in applying this to your own work, definitely check out the paper. I've worked with it quite a bit, so happy to answer questions about that specifically. But I do just want to give folks an idea of when we do like representation learning, we're learning a low dimensional representation. We have specific analysis, like goals in mind. There are different constraints that methods may have that are specific to our domain that we can then kind of say, here is a pattern that's supported by our data. There are some constraints during this training process such that some of these will tell us about things that we know are of interest to us. And then we can do things like look at differences. You can go ahead and read the model in. Plier is going to give us information about the association between a latent variable, which is going to be this pattern that it learns, this new feature, and an input gene set, which is often telling us something like cell type, what kinds of genes tend to be highly expressed in a cell type compared to others, or other kind of signaling processes that we may find. So in the interest of time, we're going to take a look at one particular latent variable. There are 58 returned by the model in this case. And take a look at the summary data frame. And what we can see in this is these are the pathways that we gave it as input. So these here are different immune cell types, this as well. And then we get some information back about the association. Typically, you can do something like pick an FDR cutoff and say this is a significant association. And then often what we're interested in working with is in this list of kind of 13 things returned by Plier. And again, you can see what we did in setup. If we run this, this B matrix is going to contain the expression values for our new variables. Or you might think of this, sometimes people call this something like a latent space. There are 58 of them that we have 121 samples here. This is kind of a wide format. Genomic data often is, so we need to get it into a long format because we want to plot the differences between our subtypes. So we run that. We should now have kind of our long data frame that with our samples, and then we have our molecular subtype labels as well. And what we're going to do is filter to a particular latent variable. Now, I know the name of this, you may have noticed. So we have the number of the latent variable and then kind of one of the pathways that it's associated with. If you are using this in your own work, one thing to know is that that's not the only gene set that's associated with this latent variable. We saw up here that there are kind of multiple. If we were to pick an FDR of 5%, there would be multiple gene sets associated with this. And let's make a box plot. So we haven't done any kind of formal test here, but we can kind of, this is another way we could potentially explore our data, where now we're looking at the LV estimate for this kind of macrophage associated latent variable. All right, these values are likely to tell us about, in this case, because it was associated with lots of different gene sets, it's probably telling us kind of some general kind of immune signature thing as higher in this particular subgroup. So some quick takeaways. Depending on the goals of our analysis, it might be useful to select a domain-specific methodology when we're trying to learn representations? Are there different constraints that we can use that will help us kind of align with our analysis goals? So often for me, I want the patterns to be interpretable from a biological standpoint, so I can kind of dig in. But kind of these combinations of genes are, you know, you can think of that often that's going to be a little more robust than maybe an individual gene measurement. It might be more informative. But we're also doing dimensionality reduction in the sense that we have these new variables, and there are a lot fewer of them in this case. We've gone from tens of thousands of genes to 58 latent variables that we could test for differences if that's what we were kind of most interested in. So I do want to point out other resources for kind of playing around with different methods that we kind of briefly made some plots for. There's this paper that's some guidelines for choosing different methods. There are lots of great tutorials that I would encourage you to check out. I'm going to run session info here and save this. And I am happy to take questions.
Thank you so much, Jackie. This was really awesome.
Thank you. Yeah, thanks everybody for spending time with us. Yeah.
We have a whole bunch of questions. So going back to the question that Alice asked on dimension reduction based on variance, do you want to give a quick summary on that?
Yeah, so often when I'm doing something like, let's say, take UMAP for an example, I am kind of most interested in the kind of overall structure in the data, this kind of overall pattern. You can imagine if there's not a lot of variance within a particular gene, that is not necessarily going to inform the overall structure, right? Things with higher variance are kind of more likely to kind of give me insight to what's going on. So often these, it can be helpful to kind of get, one thing I will say in these data in particular, there may be things that you're not reliably measuring as well that will have like low values overall. So this is not, there are kind of other ways to go and reason about that, but that's just a part of these data is sometimes you're getting rid of things that are, you are unlikely to reliably kind of detect or measure, or however you want to think about it.
Okay, and then kind of a beginner question. So the visualizations that you showed, who would be using these visualizations? Who would be using them? And what for?
Yeah. So I will say, certainly PCA is one thing when I get a new data set that I will do. And it's kind of for the way I kind of think about it is like, what's the big signal? So I think the example that I keep coming back to, and that's because often as like myself as an analyst is what I'm kind of looking out for, are things like, is what is captured in the first principle component, for example, informative about something that it doesn't necessarily speak to my scientific question. So something like when the sample was prepped, or who prepped it, what kit was used, something about that I'm like not particularly interested in kind of digging into, or necessarily like that was what I was after when I set up my experiment. Versus, so let's take cancer for an example. Cancer is often, certainly for adults, called like the disease of a genome. So we expect the transcriptome, the thing that we have essentially a readout of, to have very different structure. So if we have a set of cancer samples and normal tissue, we like know that the patterns of expression in those should be pretty different. That's not always true, but like often that is like a big signal that will kind of hit us in the face when we do something like PCA. So I think, but you can imagine you want to look at kind of groups of things that are not necessarily tumors. Like maybe you have a bunch of features for a customer, say, which, right, these are not kind of gene expression values, but there are kind of other ways to look at it. So often my question, because of like my deal, is what's the big signal, and is it something that I am kind of worried that I measured but it was not after. That's like one of the first ways I use this, if that makes sense.
Yeah, kind of as a sanity check.
Yeah.
Awesome. Let's see the next one. So regarding the discussion on consensus clustering, what is the metric used to make the clusters? So what is analogous to the different distance metrics used in k-means?
The answer is it depends. I use Pearson correlation, which kind of commonly used. I think that's the default for the method. It's commonly used for like these kind of data, but it you have multiple kind of distance options at your disposal. And then there are different, you can use different kind of algorithms as well. That particular example used hierarchical clustering, I believe. But yeah, there are different, you can change that, certainly. There are different options available to you.
And that depends, choosing which option depends on what you're after or what would make most sense for your research question.
I would say so, yeah, I agree with that.
Got it. Okay, let's see. What about some of the other options for the UMAP function? For example, PCA equals 50. So before we scaled and centered the data for the PCA, but what about for UMAP?
So I think it should scale, and I believe that like setting PCA to some integer will do that by default. But yeah, I would say often when you're like picking number of principal components, the like variance explained, the kind of elbow plot kind of point of view for that, you could use here as well, where kind of when you see a drop off in the variance explained, you could use that number of principal components as input to UMAP. 50 is just, again, a rule of thumb, which may or may not be kind of applicable. I think people, I am definitely not an expert on kind of these non-linear methods, but people say that sometimes doing principal components analysis first will kind of sacrifice some of the local structure. But I am by no means kind of an expert on the kind of nuts and bolts of those methods.
Gotcha. All right. Let's see what else we got. So once you have defined your reduced data set, was there a step to model a predictor?
We did not cover that, but you certainly, people certainly do use that in this way. Yeah, one thing, to go off on a little bit of a tangent, is one thing I'm kind of interested in is whether the latent variables learned by Plier are a little bit better for prediction than the original features. If I were to try and predict something like subtypes, an example, but and is that particularly true if they're associated with gene sets? So we didn't cover that, but that's something you could do.
That'd be pretty cool to explore.
Yeah, so here's what I think. I've tried this a little bit and it didn't work and it broke my heart, to be honest. But that could have been the function of the particular data set at hand. But I am kind of curious if, particularly if we're looking at trying things to be generalizable between different gene sets, if there are latent variables that are associated with particular coherent biological processes that we might expect to happen in different kind of cohorts or biological settings, if you will, if that helps us kind of more than the raw features. They're not raw, but are kind of original gene by sample input space.
Yeah, sounds like that would be an area to really explore further. That sounds really cool.
An open research question. If you do it, I would love to hear about it.
Let me see. I only have a couple of other questions. So is global versus local structure specific to these data? Or are these universal terms to this type of clustering?
The second one. So that is one thing that I would say is very interesting to play around with, and as you're trying to like build your intuition around these methods, and folks in kind of these different other resources do this well, is they'll show you basically random noise, and then if you do t-SNE on it, sometimes it looks like a pattern. So that makes me a little nervous. So where you like don't necessarily expect there to be local structure, right? These kind of clusters, and then there are. So just something to be aware of. If you're going to use these, I would definitely kind of play around with different tutorials, certainly. And I don't know that I have I'm still kind of on my journey for building an intuition for these methods myself. So.
One more generic or more general question. So you use Plier to identify the latent factors in gene expression data. Do you have any recommendations for other methodologies or packages for identifying latents in other data sets in general?
I would say that I think it depends. Specifically with packages, I don't know that I'm the best person. Maybe somebody else might be better suited for the packages question specifically. But I think this might be a good place to start for that kind of question specifically. It's in R, so.
That's wonderful. And we can access this, these markdown files and everything in your GitHub, right?
Yep, so the workspace will go away, but the notebooks will still be available on GitHub. It will persist.
That's awesome. And then one final question, just out of curiosity, why did you save the session info?
Oh, so I like to know the versions of the packages for kind of reproducibility reasons. This is actually the question that came in earlier when you're working across with different different members of your team. People might have slightly different kind of environments, and sometimes if everybody has their session info kind of printed out, that can be helpful for identifying what's going on. Like if there was a kind of a major update between different packages, that can be really helpful for diagnosing a problem. So I highly recommend putting that at the end of your notebooks.
Yep, that helps a lot, especially if you want to go back and check what packages were used to be able to run it, because you run it later and then you run into issues of, oh, this doesn't work anymore.
Do yourself 6 months from now a favor and print your session info. That's
Yep. All right, so this brings us to the conclusion of the workshop. It's 8 o'clock, so I don't want to keep everybody much later. So thank you, Jackie, for this wonderful workshop. This has been phenomenal. We will publish the workshop on our YouTube. We will also write a summary on our blog, so stay tuned for that. Although that might take a little bit longer. And hopefully everyone will join us for our September, October, November meetups, which are coming up and which we will publish on our meetup account. So stay tuned for those. Thank you, everyone. Thank you, Jackie.
Thank you.
We're looking forward to seeing everyone next time.